Okay, let's get started today. Sorry, the machines are wigging out again on me here, but the recording should be working. Yes, good, hopefully. That's the plan. Nobody has told me what the major problem is with either of these models yet. Great way to study for that thing that's happening on the 1st of February. So there are no biology seminars happening tomorrow, but in the following couple of weeks we'll have some job candidates, most of whom are going to be talking about molecular and cell biology. So these should be really interesting seminars, etc. So today I want to just finish up really quickly with some of our chromatin stuff, and then we'll move on and talk about replication. A couple people have asked me about what's going to be on the exam. What will be on the exam is what we cover in the next 2 lectures, not necessarily what's in the syllabus. So there are a couple of changes also relative to the PowerPoint, which I uploaded here because there's stuff that I think is probably more important for us to get through at this point. So as I mentioned last time, one of the really amazing things about the nucleus and chromosomes has to do with the fact that the nucleus is not just a bag with everything sort of floating around inside it, all the different chromosomes. It turns out the chromosomes have very specific locations in the nucleus. Now, it's not the same for all nuclei, but if you just take a nucleus and use exactly the same fluorescent hybridization probes that we used for that rainbow karyotype when we were looking at karyotypes a couple of lectures ago, and just look at a nucleus, they're spread out all over the place, but they're not overlapping with each other, and they're in very distinctive locations. So for instance here, fortunately they've got nice labels, chromosome 3 is over on this side, that's this guy, and the other copy of chromosome 3, because of course it's a diploid nucleus, is over here on this side. They're very localized where any of these chromosomes happen to be, and that's all having to do with the nuclear matrix, where these things are actually sitting inside the actual nucleus. So that's one aspect that I wanted to emphasize here. The other one is that, yes, they're present in very specific locations, but it's also still pretty dynamic, which is pretty amazing if you consider how ridiculously compacted each of those chromosomes is in the nucleus. And we talked a lot about that last time, nucleosomes, 30-nanometer filaments, extra compaction, etc. So basically doing exactly the same thing as we had on the last slide, only looking at one of the chromosomes as opposed to all of the chromosomes. You basically get an image that looks like this up at the top here. If I can get my pointer to work, here we are, where 2 copies of the chromosome are kind of on different sides of the nucleus, just rotating this around. You see the same kind of thing. But what's important here is you see these little red dots. These little red dots are, again, a fluorescent probe hybridized to a particular part of your genome, but now that's being either expressed, i.e. actively transcribed on this side, or not being transcribed over here on this side. And what you can see is these transcribed genes are way apart from where the rest of the chromosome is. So the chromosome is here, and these transcribed genes are a long way away. And what that means is, again, this is a really dynamic system. And particularly important is that euchromatin, heterochromatin that we talked about in terms of modifications mostly of nucleosomes. So heterochromatin is compacted. You don't have any kind of gene expression. Euchromatin is not compacted. And what we sort of skimmed over at the end of lecture last time is that where these expressed genes are usually in loops away from all of the rest of the chromosomes. And so that's a really important point in terms of this. And what that means is probably most of the transcription, and as we'll see later, replication, is happening just in a few very focused places in the nucleus. And that means that these DNAs are somehow getting transported where the proteins are, as opposed to the proteins scooting along the DNA. And that's something that we'll see as we move on today and talk a little bit more about replication. But everyone's, of course, wondering what happened to the clicker questions, because we didn't have them from last time. And that has to do here with change in position of the nucleus, expressed genes in the nucleus is mostly thought to be due to diffusion, dissociation of histones from the DNA, movement to expression factories, formation of lamp brush chromosomes, formation of the nuclear pore complex. Sorry, it's a little hard to see here. We're counting down 45. $30. $13, $10, $5, okay, let's get these results over here. We seem to be about 50-50 divided here, more or less, between the change in position of the expressed genes in the nucleus is mostly thought to be due to diffusion. Nobody likes that. Dissociation of histones from the DNA, movement to expression factories, formation of lamp brush chromosomes, formation of the nuclear pore. Nobody liked the nuclear pore complex, although that's where some of the matrix things are associated. So that could be an answer in a different universe. It's not in this one. So basic question has to do with the dissociation of the histones or movement to an expression factory. It is the movement to an expression factory. That's where you have the clusters of these proteins. It's the DNA which is moving. And so we will also look much more at this later on, but histones are almost always still associated with the DNA, even when you're undergoing replication, transcription, etc. So we'll talk again much more about that later on. Okay, so quick review what we talked about in terms of chromatin. We talked about nucleosomes. I'll be again just a second. The 30 nanometer filament, which is how those nucleosomes are packing together with each other, probably because they're relatively flat and then can pack on top of each other, and it's those tails that are involved in really helping that structure to come together. Topology, this is an older view slide. We'll talk about that today. Epigenetic inheritance, that's all of the modifications to the histones. And those modifications of histones can be maintained how? After replication, modifications lead to more modifications. And so that's the positive feedback loop. So if you have a modification, then a read-writer complex will come down, bind to that modification, and modify the nucleosome right next to it, and keep doing this until you get to a barrier. Heterochromatin is just the compacted, non-active chromatin. Histone modification leads to that kind of switch. Alternative histones we talked about really briefly. Those are the ones that can be shuttled in and out of the nucleosome complex. Localization we just talked about, and then the metaphase compaction last time was condensin and cohesins. Yes, your question?
So just clarification on the last.
Yeah, on the clicker?
The histone dissociation, that just accounts for nucleosome breathing, right? The momentary dissociation from the DNA.
Okay, well, let's go back and look at this here. So dissociation of histones is not going to have anything to do with that movement. The DNA can certainly move around while it's associated with histones. So that's not, you don't have a complete dissociation of the histones in order to get that. And it's more, again, this is more about the change in position is the question, much more than the actual expression process. So other questions about chromatin? Wanted to really briefly mention that most of our genome is junk. The main message here, and we've talked about this a couple of times already, is that in terms of protein coding sequences in our genome, it's only about 1.5%. That is a number that I do expect you to remember. How big is the genome, haploid genome for humans? Around 3 billion. That's as close as I would like you to remember, but that's another number that I think would be very good to remember. We'll talk much more about some of the other things, but being virocentric as I am, I wanted to mention here the 8 to 10% of clearly viral sequence that's present in our genome. So actually more viral than human in many cases here. We can make arguments that these other repeated sequences here, the line elements and sign elements, we'll talk a lot more about this when we talk about transposons later on. These may or may not be derived from viruses. And so if they are, then about 45% of our genome is viral, as opposed to the 1.5%, which is actually making proteins, which is still kind of mind-blowing to me. There are a lot of other repeated sequences, and this is actually should be a big question mark over this yellow region here, because these are the ones that we don't actually know how long they are. In fact, that's partly why I don't expect people to remember the exact number of bases in the human genome, because we don't know. And it turns out there are actually variations from people to people in terms of the number of actual bases which are there. And then sort of the even bigger question mark has to do with this dark green section here. Non-repetitive DNA spread out through the genome. We know there's a lot of it there. We really don't know what the vast majority of it is doing. Sort of why is it there? Is it just left over from evolution? Is it important in terms of regulatory sequence, et cetera? So very interesting question. The other thing to emphasize here is the introns, this pink section here, relative to the actual protein coding sequences. There's a lot more intron than there is exon or protein coding sequences. So 3 billion base pairs, we got 2 copies, makes about 6 billion in terms of the diploid genome. And we've got trillions of cells, but we start out with 1. And so how do we do that? Well, the first thing you need to do is replicate your genetic material. So this is our overview in terms of DNA replication. Going to start out talking a little bit about mutations. Mutations happen. It's something which is, in some ways, a problem because, yes, mutations can lead to problems like cancer, for instance. But if we didn't have mutations, then we wouldn't evolve. And so we would still be slime. So it's that interesting balance between having inherited changes that happen. And in fact, every time our cells undergo replication, there are a couple of changes that happen. And so maybe all that non-coding sequence in our genome is actually just a buffer so that we can have all these changes and not have too many different changes that happen to our protein coding sequences. It's a distinct possibility, and some theories have to do with that. Semiconservative replication, hopefully this is something which is not new to any of you, but it was not clear at all when people were first looking at DNA and when Watson and Crick came up with their structure and how this could actually work. Then we'll drill down a little bit deeper, talk about the mechanism of replication. This has to do with DNA polymerases and how they add on individual nucleotides to growing chains, and that process. It's actually really pretty amazing how well the DNA polymerases do in terms of putting in the appropriate nucleotide. And then we'll look at all the other proteins that are involved in this process, particularly the sliding clamp protein, which is absolutely critical for processivity. Processivity mean anything to people? Yes? No? Huh? We'll talk more about it. And then talk a little bit more about the replisome. Molecular biologists love to add zome to the end of everything. The nucleosome we've already talked about. We're going to talk about the ribosome. These are multiple protein complexes and very often proteins plus nucleic acid. And so it's just a complex of macromolecules. And then if we get a chance, we'll talk about mismatch repair and supercoiling, but given my experience with this, often we never get there. So first I want to talk about mutation. Again, mutation, as far as we're concerned here, is just a change in the genetic material. And that's, of course, DNA for all cellular organisms. Turns out that it's about 1 in a billion per replication rounds. And again, getting back to that number I expect you to remember, that's per haploid genome. Each time our haploid genome gets replicated, there is more or less 3 changes that happen in the genome every replication cycle. If you think about the number of replication cycles going from a fertilized egg to something like this, that's a lot of potential changes. And there have been some back-of-the-envelope calculations that basically said, even before we knew what the coding sequences were in the human genome, just because we knew what this mutation rate was, that max 50,000 proteins as far as the human genome was concerned. Because if there were more than that, with sort of average size, you would be making mutations in each of these particular proteins every time that you replicate it. So probably it wouldn't be a possibility. Most mutations are eliminated quite rapidly, particularly if they happen to be in an important gene. And this is actually very useful. Unfortunately, in a few cases, as we all know, those mutations end up causing an uncontrolled growth phenotype, which we also know as cancer. There's a cancer biology course that Dr. Singer teaches. Highly recommend it if you want to get more into that. We're going to have much of a chance of talking about that here. Remember we talked about the crazy karyotypes in cancer cells? That's well after you have some of these individual mutations. But if you had no mutations whatsoever, again, we'd still be slime, because it does provide material upon which natural selection can take place. Now, most of the time for the next couple of lectures, actually until after the midterm, we talk about mutations as individual changes to individual nucleotides, but mutation in and of itself is a much larger issue. It can be extra nucleotides, it can be taking away nucleotides. It can be translocations, like we talked about when we talked about karyotypes. So there are lots of different ones which are there. So our semi-conservative replication. This was actually predicted by Watson and Crick in their paper, which I know all of you have read because there'll be a question on the midterm on it. And basically, what they said is diagrammed over here, and that is that you've got these 2 strands that, again, Watson and Crick DNA, they come apart, and each of those strands is copied. Now, there are a couple of interesting things about this. The first one, okay, let's just start with a little bit of overall labeling here. The old strands are labeled in red. The new strands are the ones that are labeled in blue. And you'll notice here, this one starts at the bottom with 3 and goes up to 5, and this one starts at 5 and goes up to 3. Because, of course, they're anti-parallel, and they have to be anti-parallel. Again, I think this is one of the real huge breakthroughs that Watson and Crick came up with, and it's not something they could have got from the data at all. They had to get it from model building. Now, if you have this kind of semi-conservative replication, and this is sort of the shrinkage of what we just looked at here, now we've got the old strands here in yellow, new ones starting out in red, and then going to green, and then eventually going to black down here at the bottom. And your prediction, if you have semi-conservative replication, in the first generation, as you replicate this, you're going to have 2 copies, but each of those 2 copies, because they're double-stranded, is going to have 1 old strand and 1 new strand that's associated with it. If you do another round of replication, you're going to have 1 double set of double strands that has an old and a new, 1 that has an old and a new, that makes 2 of them, just like you had back up here, and then 2 new ones, because the new strand is going to get replicated, and so on and so forth, as you go through multiple rounds of replication. Now, the problem with this, and this is actually part of the problem that a lot of people had with Watson and Crick structure in the first place, is somehow you've got to get these 2 strands apart. That's hard. And that's probably why, no one really knows, Linus Pauling's model actually had the bases sticking out on the outside, because that's where your information is. You want to be able to access that information. How do you pull these things apart? It turns out it's really hard
to pull apart DNA, and the replication process, the most energy you need, actually comes from the helicase activity, which is pulling the two strands apart. But they had no idea what this was at the time. So there were a number of different models for how this was going to work. The three main ones were this semi-conservative replication, also they call conservative replication. Conservative replication is actually what most people thought at the time. You remember when we talked about major and minor grooves? All the information that you need to know what that base pair is in the major groove of DNA. You don't need to pull them apart. You can actually leave them together. As long as you've got some adapter that says, hey, this is this sequence over here, we need to make a new sequence over here. And so in this theoretical process, the two strands actually always stay together. And so what you'll have in your first generation, is a mixing this back and forth here. So in conservative replication, you end up after the first generation having one double-stranded set of DNA that has both old and one that has both new. And then in your second generation, the same one is being conserved and three new ones. Now, this is what, again, at the time, a lot of people thought was going on. There's a third model that they were looking at the time, which basically says you copy pieces of your DNA. And this, again, hypothetical situation turns out to be extremely true for DNA repair. So when we talk more about DNA repair, again, after the midterm, we'll see that this kind of replication, dispersive replication, which is where you copy little pieces here and there, turns out to be exactly the way that many kinds of DNA damage are actually being repaired. Here, just like you would have in your semi-conservative replication, you end up with mixed DNA populations. Half and half, new and old, only now new and old are actually in the same double-stranded pieces. And in the second generation, you would have also mixed, you're getting more and more of the new DNA relative to the old DNA. So these were the three models that were present basically at the time of people trying to start to do experiments to look at this. And so I'm going to switch over really quickly to some of you may have noticed this book here that I brought with me. Is the light? There it is. There's a lamp. So Meselson-Stahl and the replication of DNA. Sorry, it washes out a little bit here. But what I really wanted to mention was that this is the most beautiful experiment in biology, at least according to this particular author, and have to show off a little bit that Frank Stahl was actually here, gave a seminar about 8 years ago, actually no, 9 years ago now. He's now at the University of Oregon, and wonderful guy doing really cool research. And so basically, the reason that this author said that this was the most beautiful experiment in biology was that they had these three really nice models, conservative, semi-conservative, and dispersive replication, and they knew exactly what the results should be before they actually started the experiment, and then did the experiment, and the results were such where it was absolutely clear what the actual answer was. So how did they do that? Basically, it was a really great combination of two different people. Again, Meselson and Stahl, Matt Meselson and Frank Stahl. Matt Meselson knew about centrifugation. Frank Stahl knew about growing microorganisms. And it was that combination that really allowed them to do these experiments. And basically, the problem was, how do you tell new DNA from old DNA? And the way that they did it was using everyone's favorite kind of modified, i.e. different isotopes of, in this case, nitrogen. Now, those of you who are up front can see, if you look at your DNA molecule, this particular one is labeled so that you've got blues for all the nitrogens. There are lots and lots of nitrogens that you have in DNA. turns out I heard from another fellow, in fact, wrote the introduction to this textbook, a fellow who was teaching basically the equivalent course to this when I was a graduate student, which again was the late Mesozoic, but he actually told us about trying to do exactly the same experiment, but using radio-labeled phosphorus instead, and he got no results because he couldn't separate the new from the old because there are a lot fewer phosphoruses, the black molecules on here, black atoms I should say, relative to the number of nitrogens, which are there. A fellow by the name of Gunther Stent, who unfortunately passed away a couple of years ago now. So the key here was having that differential label between old and new, and because you have a lot of nitrogens, you could use a lot of nitrogens. E. coli grows really well on nitrogen, and you can just shift from growing your E. coli on N15, precursors for nitrogen, to N14, so going from heavy to light. Then you can separate the DNA from the cells, and that's when Matt Meselson comes in and did his equilibrium analytical ultracentrifugation. Just basically separating things on density. You had a question in the back.
Yeah. So the piece detections for isotopes of different molecules, they can't just detect one isotope, one molecule. It has to be a certain concentration of it. Like you said, there was a lot more nitrogens in the DNA molecule, so you couldn't find a phosphorus isotope, but is it because it's sensitive, or the equipment back then wasn't as sensitive? Or?
Yeah, so the question basically here is why nitrogen versus phosphorus, which I thought I explained to them, getting a enough job of doing so. But yeah, you have to give a universal label to new versus old strands, and it has to be a large enough difference that you can separate it with this centrifugation technique.
Yeah.
And so you have to replace lots and lots and lots of phosphorus or lots and lots and lots of nitrogen. And you have to have enough that there's a difference in density that you actually can separate using this analytical ultracentrifugation. And as we'll see when we look at the data in just a second, that the difference between the two is not very much. And even though you've gone from 14 to 15, which is a pretty large percentage change, rather than phosphorus, which is P32, versus normal isotopes, which are 30 or 31, and you've got a lot more of them. And that gives you enough of a difference in density to be able to separate. What is this equilibrium analytical ultracentrifuge? It was, at least at the time, about the size of a wall. And so this is Matt Meselson in front of his, as they say, the big machine. To give you a bit of an idea here, this is the rotor, which was spun at very high speed, and then basically a UV light was shone through this little hole in the centrifuge, and then it would expose film on the other side. UV light is absorbed by DNA, and so you'll have some areas which are absorbed and some areas which are not. And so the data which they got were these. You start out with your heavy DNA. It ends up here and gives you this band here. After 0.3 generations, or 0.7, or 1 generations, you'll notice that you end up with one band. And then after a few more generations, you end up with 2 bands. And as you keep growing, this lighter one gets heavier and heavier and heavier. Well, it's not heavier and heavier. Darker and darker and darker, which means there's more and more of it. So, what does that mean? If you look at what they expected, which is over here, starting out with your heavy or dense DNA, in a semi-conservative process, you expect to see something which is half and half, and then something which is light. If it was a conservative process, you'd expect to always have your dense DNA, and then have some of the light DNA. If you have a semi-conservative, you just have more and more of the light DNA. And so, what did the data look like? first one, the semi-conservative process, because we have heavy down here at the bottom. Sorry, pointer is here somewhere. Heavy here, slowly moving to an intermediate, and then a light. And you'll notice these things are very close together. It's actually really hard to tell apart. And so that's getting back to your point about why nitrogen versus phosphorus. And in fact, what Gunter Stent, again, probably had his graduate students do this, and he saw just one line. So he didn't tell him anything. Okay, so make sure everyone's clear on how this experiment actually works. If DNA replication were conservative instead of semi-conservative, Meselson-Stahl would have expected to see how many bands after 3 generations of growth. Okay, feel free to discuss this. Those of you who look at my old exams will notice that this is a question I love to ask. Yeah. 30. 2 and 3 seem to be people who are, actually one's pretty similar too. 2 is the correct answer. What are those 2 bands? They're heavy and light. Those are the only things that you see. You don't see an intermediate band. And so, very important that that one has completely disappeared. What that says is, you really have pulled those 2 strands apart. Now, how those 2 strands got pulled apart, Meselson-Stahl actually had absolutely no idea. And in their paper, actually they don't even show those nice wrapped strands around each other. Just have blocks, like rectangles, that they talk about for them all. So 2 rectangles separate, and then those separate again, or they don't separate, relatively speaking. Okay, so what does this? What does this making of the new DNA? What does the making of the new DNA is a, surprise, surprise, DNA polymerase. Yeah. Oh, sorry. Oh, so the question is, do you have enough heat to separate the 2 strands relative to each other? So the vast majority of this work is done on E. coli. And so E. coli grows optimally at 37 degrees Celsius. At 37 degrees Celsius, your DNA is completely bound together. If you think about the melting temperature that we talked about way back when we talked about the reannealing of DNA, usually the melting temperature for E. coli DNA is in the order of 70 to 80 degrees Celsius. So way too hot for the organism normally to be able to deal with. My favorite organisms grow at 80 degrees Celsius. So, yes, no, you actually have to have a way of pulling those 2 things apart. And we'll talk about that a little bit later on. But first I wanted to talk about the actual machine which does the job, the DNA polymerase. And this is almost all work of Arthur Kornberg, father of, the only father and son Nobel Prizes that I know of. There's a mother-daughter Nobel Prize. You know who that is? Way earlier than the Kornbergs? Curie. Marie Curie and her daughter, whose name is escaping. So, Arthur Kornberg was working on this. I don't really like people to remember dates. It's not a history of science course. But I just wanted to point out here that the discovery of these enzymes and also Meselson and Stahl's experiment are very soon after Watson and Crick's structure. So the early to mid-1950s were a really amazing time in terms of molecular biology. 53, Watson and Crick's paper, Meselson and Stahl, which was 58, the DNA polymerase, 57. So lots and lots of people who are working on all of this, really interesting genetic material, even Avery's stuff in the mid-40s, in terms of DNA being genetic material. Hershey and Chase, 52. So all this is happening more or less at the same time. So what Arthur Kornberg was doing was basically, again, going back to the coli we were talking about before, he being, of course, his students and postdocs, purified this activity that they found in E. coli that could take DNA and make more of it. And to make a long story short, they figured out that it needed 2' deoxynucleotide triphosphates to be able to make this, so those are your precursors. Also needed a template, so it needed a single-stranded piece of DNA. They actually used a viral DNA, so you didn't actually have to pull the 2 strands apart, which is very convenient. They also used heat, though, in the same process to do this. And what was really kind of surprising is they always needed a primer. They needed to start with something. And that primer turned out to be, you had to have a free 3' OH. So all of these DNA polymerases have to have something to start with. And this is one of the big puzzles that people didn't solve for quite a long time, actually, was that all these DNA polymerases could extend an already existing piece of DNA. But they couldn't start by themselves. We know now later that an RNA polymerase, which says that RNA polymerases can do this, DNA polymerases can't. The other thing that they found was that this particular polymerase had to add at a 3' OH and always went in a 5' to 3' direction. So your chain is growing here, 5' to 3', which is the opposite orientation relative to the other strand. Because, of course, these are anti-parallel strands relative to each other. Rumor has it that Arthur Kornberg kept going back to his students and saying, you need to find the other polymerase. You need to find the one that goes from 3 to 5 rather than 5 to 3. I don't know why was that? Well, because when you pull apart 2 strands, you remember one's going from 5 to 3, the other one's going from 3 to 5. How does that work? Well, we now know that it's not only semi-conservative, it's semi-discontinuous replication with Okazaki fragments. So actually, all of these DNA polymerases go from 5' to 3'. How do they do that? They take nucleotide triphosphates, again, the deoxynucleotide triphosphates, which were what Arthur Kornberg's group needed to have this. You have a primer here with a 3' OH at the end. Your nucleotide triphosphate comes in as long as it's the one which is appropriately base paired, and we'll see how that happens in just a second. Pyrophosphate is released. What's pyrophosphate? It's just these 2 phosphate groups attached to each other, and then you have hydrolysis between those 2 by a pyrophosphatase. It's this 2-step reaction which makes polymerization basically irreversible. If you think about this from an energetic point of view, particularly an entropic point of view, adding free-floating nucleotides to a really long chain is entropically really hard to do. And so the only way that you can do this is to have coupled reactions with these high-energy bonds, which are getting hydrolyzed, and not just the first one, but have 2, these 2 enzymatic reactions back and front. So the polymerase just does the first one. It's a pyrophosphatase, separate enzyme, which does the second one. What does the structure look like? This is a model of the structure here. Ribbon formed out at the bottom. Blue is the DNA polymerase. Gray is DNA with the site where you're actually going to have the addition. Again, that triphosphate being added to the 3' OH is right here down what they call the palm of the DNA polymerase. We always talk about palm, thumb, fingers, etc. Basically, I'm not sure how you see it, something that looks like this purple thing down here at the bottom, to look like a hand structure. But again, that's what people talk about. There's a thumb, which is this piece. Come on, my pointer to work here. Thumb is in this part of your protein here. Yeah. Okay, the thumb's on the right-hand side here. sticking up. You can sort of see that sort of coat hanger structure, and then the fingers sort of wrap around your DNA, and it's in that wrapping process where you're holding the DNA and basically checking to see that you have the right nucleotide which is coming into your active site. The active sites we talked about with enzymes, all the thumb, fingers, etc. are all there to just get the appropriate side chains to the right place so they can catalyze this enzymatic reaction right there. And then this active site requires 2 metal ions, they're slightly different for different polymerases, and those have to be held in the right place by the appropriate amino acid side chains. And this template is a little bent. You'll see why it's bent in just a second here. I'll have to move this slide. Oh, well, never mind. We'll talk about why that is right now. Go back and just one second. The polymerase itself has these alpha helices right here with all of these amino acid side chains sticking out of it. And basically what this is doing is making sure that the right base pair is formed here. Now, how does the polymerase know, to totally over-anthropomorphize this, that the right base pair is there? What the polymerase does is it basically folds down this alpha helix on top of the base pair and says, is it a normal base pair? Is it something which is nice and flat, that's going to interact with particularly this tyrosine here, which has an aromatic group on the side? Does that flat down right on top of your DNA molecule? And if you look at Watson and Crick DNA, those proper base pairs, the ATs and the GCs, are in a really nice location and nice and flat. If, however, you've got the wrong kind of base pair which is there, the wrong nucleotide is there, it's not nice and flat. And so, if it's not nice and flat, you don't have this flopping down of the tyrosine, and what happens then is you undergo what's called the proofreading reaction. And what that is it cuts out the nucleotide which had come in, and in some cases actually now incorrect structure. And we'll see this is happening again and again and again when we talk about DNA repair. It's not so much the actual base itself, it's the structure which is being detected. So if you've got an incorrect base pair which is formed, it changes the structure of your DNA. And it's that structure which then says, basically, again, to over-anthropomorphize, hey, polymerase, you just put in the wrong thing. That was a bad idea. It's time to get rid of it. And so this wrong nucleotide, which gets put in, basically is shown here. Look, we've got the wrong nucleotide that's been put in. We have the wrong structure. Then there's an exonuclease, which comes in and chops that off. Now, this particular exonuclease is backing up and chopping off the one which you just put in. What direction is that nuclease going? Polymerization is from 5' to 3'. If you're backing up, it's going to be 3 to 5. And so it's a very specific nuclease. It turns out to be in the presence of most of these DNA polymerases. I'll talk about different ones later. It's actually part of the DNA polymerase. It's actually a separate domain of the DNA polymerase which has this particular activity. Once you have this exonuclease, and just a quick reminder, I've talked about this already, exo means coming in at the end, endo means coming in the middle. We'll talk about endonucleases right at the end of the class. Exos are just coming in at the end of your DNA molecule. So the exonuclease just chops off that last one. Now you have a 3' OH, which is appropriately base paired to your DNA, and it can be used to restart replication. The polymerase is actually right there, and so it can go forward here. Turns out this process only works with 5' to 3' polymerization, and why that is outlined here. So if you had incorrect bases which were added here at the 5' end, remember 5' says all these phosphates, 5' triphosphate. All the nucleotides have 3 phosphates hanging out at the 5' end of each of your nucleotides. If you had a new nucleotide that was put in, it was the incorrect nucleotide, that would then get chopped off, but now you don't have a triphosphate here that you had up here. Remember, you started here, 5' is always 3 triphosphates. 5' triphosphate adds the next one, you lose your pyrophosphate, that gets cleaved by pyrophosphatase. Now you have a triphosphate at the 5' end. If you had this polymerase, it would come along and continue to do this. But now if you have proofreading, that chops off your triphosphate. Well, if you don't have a triphosphate, where do you get the energy for your polymerization? Unless you happen to have 3' triphosphates, which would be the other way of looking at this. So the fact that you've got your triphosphates attached to the nucleotide and then are doing this adding process means that you have to have a 5' to 3' extension and not the polymerase that Arthur Kornberg kept telling his graduate students to look for, this 5' to 3' process. So the DNA polymerase itself has these activities. Again, the polymerase activity, the exonuclease activity, both of these are present in that DNA polymerase. Now, because of that, I'm sorry I got these slides a little bit out of order here. What that means is, if you have to replicate your DNA, you have to replicate it always from 5' to 3'. So how can you do that? The only way you can do that is by having extra little pieces which you put in on what's called the lagging strand. So leading strand, lagging strand. Leading strand is when you pull the 2 strands apart. One of those is going to be a template, so you can go from 5' to 3'. You're perfectly happy. Your polymerase is cruising right along. On the other hand, that opposite strand, you have to now make separate little pieces. Those pieces get attached to each other. Those pieces are now called Okazaki fragments. Reiji Okazaki in Japan showed that these happen. Actually, he thought it was both strands were being made in these little separate pieces. But now we know that one strand is mostly being made in a continuous fashion. This will be your leading strand. And then the discontinuous fashion on the other strand. So we have semi-conservative, semi-discontinuous replication. So lots of semis, half and a half. Okay, so sorry that slide ended up out of order there. Okay, any questions about this before I ask my quicker question? No, sure. Go back. So how does the polymerase activate itself? I'm not sure I completely understand your question, but the energy that the polymerase is using for doing this extension process all comes from that nucleotide triphosphate hydrolysis. So the new nucleotide is going to get added. And so all that energy is from those phosphate, pyrophosphate. Other questions on the actual activities here? No. Let's do a quicker question. It's so much fun. DNA polymerase 1 does not polymerize from 5' to 3', probably because the DNA polymerase cannot move from 3' to 5'. The DNA polymerase cannot move from 5' to 3'. The energy required for polymerization comes from hydrolysis of the DNA, because proofreading would be impossible. I should have put none of the above for E, because someone picked it. I'm not quite sure why they did. So there were only 4 options here. I guess you really are not sure. So answer that most people like is, the energy required for polymerization comes from hydrolysis of the DNA. We just talked about that. Where's the energy coming from? The energy comes from hydrolysis of what?
Nucleotide.
The nucleotide triphosphate. It's not coming from hydrolysis of DNA. Okay, so what does that leave us with? Movement of the DNA polymerase from 3' to 5' or 5' to 3'. Immediate question you should have asked during a midterm with Dr. Stedman, do you mean relative to the template or the non-template strand? Which I didn't say. Which I should have, but I didn't. So that really only leaves us with one answer, right? Hey. Proofreading. So it's proofreading, which is the probable reason that you don't have that other polymerase activity in the one that
Move the box up a little so I could see the answer.
Pardon?
Could you move the box up a little so I could see the answer?
Oh yeah, so because proofreading is impossible, or would be impossible. So hang on. It's a little challenging here, but it'll be in the recorded part as well. Yeah, because proofreading would be impossible. Okay, well, a couple of people have asked me about this as well, just as a quick reminder. Yeah, what happens if I don't get all the flicker questions right? Like this one, which obviously did a crummy job of explaining. My bad, not your bad. What I do with all of the exam scores, all the clicker questions, I normalize to the high score. And that means that probably no one's going to get 100% of all of the overall clicker questions. But if the high score is 80%, that means it's 80%. So you can have missed 20% of the clicker questions and still have the high clicker score. So it's kind of a way that I can normalize myself for bad questions or doing bad explanations. Okay, so briefly talk about Fidelity and some of the other proteins that are involved in replication. First one was what we just talked about. Oh, when I say fidelity, what's fidelity mean? Fidelity just means that you're putting in the right nucleotide. And that right nucleotide actually stays in after the whole replication process. So far, we've just talked about the polymerase. And that's the thing up here at the top, 5' to 3' polymerization. So 1 in 10 to the 5. So that's 1 in 100,000 bases get put in improperly. That's probably mostly because of polymerization. Remember Watson and Crick were talking about how they had amines and ketos versus enols and immunos. That happens in nucleotides. And if it turns out that one of it happens to be in the wrong state, you actually can get a base pair that looks actually pretty reasonable. And in that process, the polymerase puts the wrong one in. That will isomerize back pretty quickly and give you the wrong structure. That's where most of the proofreading comes in. So between proofreading and the actual enzyme itself, mostly just flopping down and checking that it's a right flat base pair, it's 1 in 10 million. But as I mentioned before, it's actually more like 1 in a billion, and in this case it's 1 in 10 billion, the numbers that they give you, in terms of actual changes. And so that other difference is what's called mismatch repair that, given it's 10 past 11, we're probably not going to get to in the rest of today, but we'll talk about that on Friday, which is very closely coupled to this replication machinery. And it's basically in the multiplication of all of those that you get to approximately 1 in a billion base pairs replicated actually stays as a stable chain. Again, 3 for each of our haploid genome replications. So what else is there at replication in the replication fork? The big problem, again, other than that fact that we don't have a 3' to 5' polymerase, is that all of these polymerases had to just have a primer to start from. They couldn't start by themselves. So what you need, surprise, surprise, again, the incredibly creative molecular biologist, a primase to make a primer. Now, this is very confusing, unfortunately. It's called a DNA primase because it primes DNA replication, but it's actually an RNA polymerase. It puts RNA onto a template. As I mentioned before, RNA polymerases can start. They put one nucleotide down onto a template and then extend from that. All DNA polymerases have to extend from an OH which is already there. And so this is the big difference here. So an RNA polymerase is shown as this blue blob here. That then puts down a couple of RNAs, these blue ones, and that then gets extended by the regular DNA polymerase, which now has, hey, here's a 3' OH. It's right next to my template. Oh, we're good to go. We can extend this. And so that process, then what you end up with is this kind of situation right here. We've got RNA connected to DNA, and particularly if you're on the lagging strand, this is true on the leading strand as well, right when you start, you're going to have these little bits of RNA, which are here. So you need to get rid of that RNA for a couple of reasons. One, it's kind of hard to replicate the RNA, but the more important reason is that RNA polymerases, unlike DNA polymerases, don't do a really good job of always putting in the right nucleotide. These are relatively low fidelity. So if you don't have a bunch of changes that happen, you want to get rid of those RNAs. How does it happen? You have an RNase, also called RNAse H. RNAse H, the H stands for hybrid. Hybrid because you've got DNA on one strand and RNA on the other strand. So RNAse H will chop up that hybrid, and then DNA ligase will link the two ends of the DNA together. All the DNA ligase does, it takes a 5' phosphate and hooks it to a 3' OH. Yeah?
So DNA primase is adding RNA, and RNA primase is adding RNase is adding DNA?
Okay, so this is, again, this is unfortunately complicated terminology here. So the DNA primase is an RNA polymerase. It puts down RNA.
Right.
The RNase takes away the RNA so that the DNA polymerase can put DNA in. And so that's actually shown here on the next slide. It's another way of looking at it. So we have RNase. So this is what's left. You've got this little RNA primer. Here's your DNA polymerase. You need to get rid of this. RNase H chews this up. And now you've got a 3' OH. Your DNA polymerase can come in and extend this. Now, interestingly enough, that DNA polymerase 1 that we talked about before, Arthur Kornberg, first one he found, so we called it number 1, also has a very bizarre exonuclease activity. We already talked about its 5' to 3', so 3' to 5' exonuclease. It also has a 5' to 3' exonuclease. It actually has two different kinds of exonucleases. And so what that means is you can actually have a DNA polymerase that sits down here, is moving from 5' to 3', so it's polymerizing from 5' to 3'. But it also has a 5' to 3' exonuclease activity. So it can actually chew up this RNA which is here. So there are two different ways of getting rid of primers. There's RNase H, and if you happen to have a polymerase, like DNA polymerase 1 from E. coli, that has a 5' to 3' exonuclease. This actually should be both. It should be 5' to 3' and 3' to 5'. What's the 3' to 5' exonuclease? Let's talk about fidelity-wise. It's a proofreading activity. It's backing up. But 5' to 3' is chewing away nucleotides in front of your polymerase, which is great for getting rid of RNA primers. It's not so good in terms of your DNA. So you've got RNase H, you've got your primase, you've got sometimes your DNA polymerase 1, but as I mentioned before, we talked about the Meselson-Stahl experiment. One of the major problems is how the heck do you get these two DNA strands apart in the first place? Well, you have called a helicase. Now, helicases probably end up hydrolyzing certainly the most ATP and arguably the highest number of nucleotide triphosphates. Basically what they do is they take one strand and pull it off another strand through hydrolyzing lots and lots and lots of ATP. Actually requires probably about, people argue about this, 6 ATPs per couple of bases which are pulled apart. And the way that this works is it's a ring which forms around one strand of the DNA, and then all of these loops in the middle here And basically these loops just sort of wobble back and forth, and in that process pull the two strands apart. And basically what they're doing is they're sticking each of those amino acids in between each of the bases and then just popping them apart relative to each other. These go very quickly, thousands of nucleotides a second, and again, they require a huge amount of ATP. Once you pull these two strands apart, do you have a question? Sorry.
Yeah, how does it get into the helix?
How does it get around one of those strands? That's an absolutely fabulous question. We'll get to it on Friday. Called a helicase loader complex, but yeah, we'll talk about that on Friday. So once you pull these two strands apart, you have a big problem. What's that problem? They love to come back together. They hybridize. You know, we talked about before. As they get the two strands apart, they can base pair with each other. This is one of the things that makes doing those beautiful different colored chromosome experiments possible. They love to come back together. So you have to have a way of stopping that happening. And again, the molecular biologists are incredibly creative, and they came up with this protein called the single-stranded binding protein, SSB. So, single-stranded DNA binding protein, it binds to, surprise, surprise, single-stranded DNA. And once it binds there, you have one that binds, and then as soon as one is bound, it actually changes its structure a little bit, which allows many, many more to bind right next to it. And that's what's called a highly cooperative process. Once you have one binding, you have binding of many, many more of these. And so this is that single-stranded DNA binding, which turns out to be really important for this replication process to take place. And as you would imagine, there's quite a lot of single strands when you're undergoing replication, because that helicase is cruising along at 1,000 nucleotides a second. So if you look at the proteins that are present inside a nucleus or replicating E. coli, one of the most common proteins is actually the single-stranded binding protein. Huge amounts of it you find in these situations. So that brings us to the last member of the major part of the replication complex. And before we talk about that, we have to talk about processivity. Now, what's processivity? Processivity is once you're onto the DNA and you're doing something, is that you stick with it. And so in this case, processivity has to do with your DNA polymerase. And your DNA polymerases, turns out they're actually, most of these DNA polymerases, the vast majority of them, and certainly all of the DNA polymerases we've talked about so far, are non-processive polymerases. And what that means is they fall off the DNA all the time. So each time they put in a nucleotide, there's a real distinct possibility to fall off and not continue. The way that's solved is through the process of what's called a sliding clamp. So instead of the helicase wrapped around one strand, now you've got a molecule, a protein I should say, that's wrapped around both strands. And those basically hang on to the polymerase, and hanging on to the polymerase allows it to stay on the DNA. So when the DNA polymerase tries to come off, it's stuck there by the sliding clamp, which is around the two strands of DNA. And it turns out it's extremely similar. It also needs a loader like you do for the helicase, which we'll talk about on Friday.